Hello again. So this video is part of Osmosis and Plant Transport, and in this part we're going to look at osmosis experiments. So the first one we're going to look at is how you can use different solutions to calculate the concentration of sucrose in potato cylinders. And the second one is using whisking tubing to demonstrate osmosis. So let's get started. Which then brings us on to this, which is a little practical involving osmosis in potatoes. So what you need is a couple of potatoes, a cork borer, and you simply just push the cork borer through the potato and get out some potato cylinders. The next thing you need to do is trim the skin off each little bit of potato. And then, once you've done that, you need to make sure that each potato cylinder is the same length. And that means that this experiment will be a fair test. The next thing that you would have to do before you put them into the cylinders here is dry them and weigh them. We then make up a few solutions of different concentrations. Now for this you can use salt or sugar. So once you've dried and weighed your potato cylinders and made up your solutions, you then put cylinders in each beaker and leave it for 24 hours. At the end of 24 hours, you simply take them out, dry them, reweigh them, and calculate the change in mass. Or, what this little video is showing you is instead of calculating the change in mass, you can calculate the change in length. So there's two different ways of doing this experiment. So the method for this one then is pretty much cut 5 potato cylinders, trim them to the same length, dry and weigh each cylinder and record the mass. Now, the reason why you need to trim them to the same length is to make sure that it is a fair test. And the reason why you need to dry them is because they might be a little bit damp, so it removes any surface water or surface liquid. You then record the mass. You place one cylinder in each solution. Now, in this one, we're going to use 5 solutions of increasing concentration of salt or sugar. One of the solutions should be water. So what we're aiming to have is 5 test tubes, each with its own solution in it, and a potato cylinder. Now, in this case, we're using sucrose solutions. That's just a type of sugar. As I said, you can use sugar solutions or salt solutions, and both will give you similar results. You then leave it for 24 hours. You then dry it and reweigh them. Again, you dry it to remove any surface liquid on the potato cylinders. And then you calculate the change in mass. Or what you can do instead is measure the length at the start and then the length at the end and calculate the change in length. So let's focus for now on calculating the change in mass, because that's a more accurate way of doing it. Because these cylinders don't actually change in length that much, it's better if we can do change in mass. So after the 24 hours and you've weighed them at the start and the end, you would calculate the change in mass and record your results in a table like this. Now what you would find is that this one should increase the most, and this one will decrease the most. And then hopefully, if everything's gone around to plan, this one should be the smallest change. So this should be in and around 0%, hopefully. And this equation just shows you how you would calculate the percentage change in mass. So for example, let's say for one of them, our initial mass was 5 grams, and the final mass was 5.2 grams. What you would do is take 5.2 minus 5 divided by 5, and that would equal, well, you've got to times it by 100, remember? So that would equal a 4% increase. Now, of course, you can do that if it's a negative value as well. So let's say our initial mass is 5, final mass is 4.7. So you still do 4.7 minus 5, which gives you minus 0.3. And then minus 0.3 divided by 5 times 100 equals minus 6%. So that shows a decrease in mass by 6%. Now, the overall point of this experiment is that we're going to use these 5 solutions and these potato cylinders to work out the concentration of the solutions inside the potato. Or we can say that we want to work out the sugar concentration inside the potato. So this bit just shows you the results you would expect, or the changes you would expect. So the one in water will increase in size. Now, obviously not that much. B will increase a little bit. C probably won't change. D will shrink a little bit. And E will shrink a lot. Now, obviously this has been exaggerated quite a bit. You might not even be able to see the changes in each of these cylinders just by looking at them. So you'd need to measure something about them. Now here we have our little table of results over at this side, where we can see the solution concentration against the change in mass. Now, in this graph, what we're going to be able to use this for is to calculate our concentration of sucrose solutions in the potato. Now, to do that, we use a graph like this. So our y-axis is split into positive and negative values, showing the change in mass as a percentage. And then we've got the solution concentration on the x-axis. So let me just fill in the scale for this. So 5, 10, 15, 20. There, that scale's better and it allows us to be more accurate. So at a solution concentration of 0, our change in mass was 19. And then we plot the rest of the points. So at 5, the change in mass was 11. At 10, it was 1. At 15, it was minus 10. And at 20, it was minus 18. Now, what we would do next is, instead of joining these up point to point, we would do a line of best fit. So you want to try and have an equal number of points at both above and below the line. So you put your ruler there, and then you draw your line of best fit. Now, the single most important point on this graph is the point here, where the line of best fit crosses the x-axis. There is no change in mass. Therefore, the solution concentration is equal to the concentration of sucrose in the potatoes. And this value is going to be 10.25. And that's where it crosses the x-axis. Now, as we can see here, any solution below 10.25 causes an increase in mass, and 10.25 causes no change in mass, and then any solution above 10.25 in concentration causes a decrease in mass. Now, the next thing to do is try and explain that. So we said the 3 things you need to say is which way water moves, how, and if water enters or leaves the cell. And in this case, does it enter or leave the potato? So, water moves from high concentration to low concentration through a selectively permeable membrane by osmosis. So the water moves from the solution to the potato, which is why the potato increases in mass. The second one, there is no change in mass because the water concentration is equal between the potato and solution. So water doesn't move in this case. Whereas in this one, any solution above 10.25 causes a decrease in mass. This is again because water moves from high concentration to low concentration through the selectively permeable membrane. This time it moves from the potato, which has the higher water concentration, to the solution, which has the lower water concentration, and it moves by osmosis. Now let's look at some potential questions with this investigation. So firstly, how would you use your graph to find the concentration of sucrose that has the same concentration of the potato? As we said, it's the point where the line of best fit crosses the x-axis. It shows the sucrose concentration that has the same concentration as the potato. The next one suggests why you should use the line of best fit rather than point to point in this investigation. It's because a line of best fit allows you to consider all the data when identifying the sucrose concentration. If a point-to-point line was used, then only the points on either side of the x-axis would contribute to working out the sucrose solution that is the same as the potato. So by doing a line of best fit, we take all our results into consideration, and we can then make a very accurate guess. Using your data, estimate the concentration of sucrose that could have the same concentration as the potato. We did that. That was approximately 10%, because that's where our line crossed the x-axis. A similar investigation could be carried out measuring the length, as we talked about earlier. Suggest 2 reasons why it's more accurate to use mass rather than length. Now, there's a couple of reasons here. The mass can be measured to at least 1 decimal point, so it's more accurate. The ends of the potato cylinders might not be even, and the cylinders could be a different thickness. The main one there is that it's more accurate to calculate mass. Why is it important to use percentage change rather than just change in mass? And that's because the potato cylinders could have been different masses at the start. It can be very difficult to use the same potato for this investigation to get enough cylinders. So therefore, there's going to be a little bit of variation between the potatoes, so it's impossible to make sure that they're all the same mass at the start. But by working out a percentage mass, it shows how each cylinder changes mass, and then you can compare them very easily. How can we make it a valid or fair test? Well, you need to make sure that in each test tube you've got the same volume of sucrose solution, the same size of the potato cylinders as far as possible, so you use the same cork bore and measure the length. You leave them in the solution for the same time, and you leave it in the same temperature. And that reason, same temperature, is because remember, osmosis is basically fancy diffusion, and higher temperatures increase the rate of diffusion. So therefore, if if we have higher temperatures, it increases the rate of osmosis. So you need to make sure that you treat each cylinder thoroughly. How could you make it more reliable? You repeat the experiment and take an average. And just because it's popped into my head, how could you make it more accurate? Or how did we ensure it was accurate? Well, you would have to use small measuring cylinders. And you could record the mass to 1, possibly even 2 decimal places. And as well as that, you need to make sure that you dry the cylinders before weighing. The second experiment then is much more straightforward. In this, it's just using visking tubing, which you'll remember we talked about in diffusion, and we said that it was semi-permeable, which means it allows molecules of a certain size through. Now, the reason why it's good is because if you got a microscope out and looked at the visking tubing, you would see that there was little pores in it. So in this case, it can act as a semi-permeable membrane because it has little pores. So the aim of this investigation is to model the movement of a substance through the cell membrane using visking tubing. So what we're going to do is add in sucrose solution into visking tubing and tie it at each end so that you get something that looks like this. You dry the outside of the tubing if necessary and weigh the tubing and contents. You leave it in a beaker of water for 1 hour. So we've got the beaker of water, we've got the visking tubing with the sucrose in it. Now, after the hour, you pat dry the outside of the visking tubing and reweigh and describe and explain the results that you see. So what you had to do was weigh it at the start and weigh at the end. So let's say, for example, at the start it was 10 grams and at the end it is 12 grams. So describing the results is pretty simply the mass increased by 2 grams. Now, how do you explain that? You explain it using these steps that we talked about. We talk about which way water moves, how, and if water enters or leaves the cell. In this case, does water enter or leave the tubing? So we've said that the tubing increases in mass. The water moves from an area of high water concentration to low water concentration through a selectively permeable membrane by osmosis. So water enters the tubing. Now looking at this diagram, here's the area of high concentration, here's the area of low concentration, so the water molecules move from there to there through the selectively permeable membrane by osmosis. And that's the end of this topic. Thank you, goodbye.